years of it's been 2 years of SARS-CoV-2, and I've been involved in helping with set up the bioinformatics pipeline, collating metadata and going through the analysis and then sending that back to the central repository in the consortium, and then submitting data to public repositories like ENA, SRA, or GISAID. We're still doing SARS-CoV-2 genomics. And so here I'm representing the average bioinformatician with practical experience, and I'm going to share that practical experience with you so you can avoid a lot of the mistakes we've made along the way. Hopefully by now you've understood the basic workflow of sample preparation, sequencing, and analysis. And I'm not going to repeat the workflow in detail here, but I will refer you to some online resources or publications throughout the talk that you can refer to later. What I will talk about is some of the important caveats that I've run into or heard from others. And this is a basic summary of everything that we'll cover. And I'll introduce these problems and talk about how you can possibly address them. I'll talk about, firstly, what you'll run into with sequencing data, and then problems you'll run into dealing with your contextual data, your metadata in your various projects. So in terms of sequencing, the most important thing to remember is that SARS-CoV-2 is a little bit different to biological agents that you've worked with. You can't use the same approaches that you've used, say, in bacterial genetics. And if you're following the tiling amplicon approach from Arctic, that introduces its own peculiarities that you will also need to take into account. So the second thing to remember is that after 2 years of doing this, most of the basic tools for exploring SARS-CoV-2 are now readily available. And you will find a lot of these, quite a few of the existing pipelines are based off the original Arctic pipelines. And you can see that link down there in the bottom that has a lot of these different pipelines you can go to. And this table here shows you a bunch of the available ones that you can use. That being said, one area is you might need to do your own bespoke analysis as well. So this is when you're doing, say, outbreak surveillance and things like that. When you're looking at a specific scenario, you might need to dig in the data yourself. But yeah, usually your baseline tools are, these are the basic pipelines you can use that will give you a consensus sequence, a mapped file that you can start to play around with to answer the scientific questions. Now, I say all of this and I'm promoting the use of existing software because there have been some horrible examples where people have just used tools that they're used to from other organisms and didn't understand what was going on with COVID, and they've made some really horrible mistakes. One of the really simple issues I've seen is that some people will try to do a straight de novo genome assembly of the Arctic amplicon data, which doesn't make any sense. For one, the reads still include the synthetic primers, so that the assembly was a weird mishmash of the viral reference genome and the sample they were looking at. In this case, it's important to keep in mind that your amplicon data does require extra filtering to mask those primer sequences before you start your variant calling. But this is all handled in the SARS-CoV-2 specific pipelines, so you should understand what those pipelines are doing and just sort of leave it at that. One of the more complicated examples is people adapted a practice that was common in human and bacterial genetics, where you map the reads to a reference, you call the variants, and then in areas where you have gaps, you backfill with the reference genome. So other times people might call variants and then alter a reference genome, and then on other times people will have a reference genome and then they'll take the mutations and sort of drop them into the reference sequence and make this mutated reference to represent their actual sample. In all three cases, this is not something that's common in SARS-CoV-2 genomics. It's something you shouldn't. If you do this for a specific project, that's fine, but don't upload these data to public databases as if they were a regular, complete SARS-CoV-2 genome. This is not what people are expecting. This is not common practice. Please just don't do this. Moving on to a more simpler matter, but something that's equally important, you need to make sure that you've explicitly removed human reads from your data. Obviously, your SARS-CoV-2 samples coming from humans, occasionally you'll get host genome that may be sequenced. and you need to remove these reads before you deposit them in most public archives to protect the host, particularly since many of the samples may have different levels of consent attached to them. So even though with Arctic you're using a targeted sequencing approach, some host does sneak through every now and again, so it's best to double check. The simplest solution is to map your reads to SARS-CoV-2 reference genome and then remove all of the unmapped reads before public release. There are other methods, like you can use Kraken and so on if you want to try that. I've got a list on the right side of all of the different tools that people have developed, some specific for SARS-CoV, some more generic, that you can also have a look at and use off the shelf, so you don't have to develop your own. One thing to consider is that the initial viral load in the sample will impact the utility of your data. If you have too little virus, so high CT in your sample, there may not be enough material to generate a complete genome, something that you can use for phylogenetic analysis. And this is something that's just a problem no matter how much you amplify it. Like the material just simply isn't there. Knowing this in advance can help you pre-filter samples, and you can determine whether they're likely to answer your research question or not. The exact cutoff of what cycle threshold to use does vary a little bit. There isn't a hard and fast rule. Once you get started with your own samples, you'll start to figure out what that cutoff actually is. In studies that we've done in our local area, we found that a CT over 32, you start to see this drop off in genome recovery, which is just shown here on this plot on the right. And where the CT value is the x-axis, and then the percentage of uncalled bases of called bases is on the y-axis. Remember that for phylogenetic analyses, you generally want over 90% of the bases reliably called. And when I say like reliably, I mean that you have at least 10 times coverage for each base for Illumina and over 20 times coverage for each base for Nanopore. These are the default thresholds that you'll find in the Arctic pipelines. You may have other research questions where samples with lower yield may be useful, and that's entirely up to you what you do in that space. But this is sort of the general thresholds that people use. This is pretty obvious, but it may sometimes seems to be overlooked. Controls matter for every sequencing experiment. You obviously want to have a negative control at a minimum, and they should also be bioinformatically analyzed to qualify the level of contamination that's introduced during the preparation and sequencing process. I have to stress this because SARS-CoV-2 sequencing has a number of peculiarities that makes this really, really important. On the one hand, you're heavily amplifying your starting material. So I mean so heavily that for Illumina sequencing, we regularly go up to 1,000 times coverage, maybe up to 3,000 times coverage. And this is to address issues that are around barcode hopping and variation in primer efficiency that can cause problems. But sequencing at that depth, you need to keep your protocol really clean, and you need to check. And the other thing that sort of makes this more complicated is SARS-CoV-2 has a very low level of genomic diversity. So even when there's a small amount of cross-contamination between two lineages, you can get very, very different results. So this is an example of the kind of a negative control versus the positive control, what that looks like. One thing I'll point out is I've put this little box in blue. This is like a danger warning thing. So this is like a portion of the negative control has reads that are greater than 10 times coverage. So that's potentially something that could be called as a base or a mutation. And if that's something that's throughout all of your samples, that's sort of a problem, because you might get some sort of weird, sort of hybrid fake thing, which will give you a weird lineage call. And there are cases in the press where people have this sort of problem where cross-contamination occurs and they do get led down the garden path and think that there's something going on, and there really isn't. Here's a range of possible errors that can happen: cross-contamination, primer, dimer, and so on, listed up here. And here are potential strategies that you can use. Feel free to screen cap this if you want to come back to it. I think the final mapped BAM file that you get out of the analysis pipeline has a lot of information, and being good with SAM tools and knowing about these different problems, you can check a lot of things by hand really quickly. And you're interested in things like dimers, or you want to see certain regions that are dropping out for certain a combination of SAM-tools depth and SAM-tools pileup and understanding cigar strings will help you a lot. And then there's a lot of existing software out there that you can use that can help you solve a lot of these problems. That's about it on the sequencing side. And I'll just talk really quickly about contextual metadata. We do have a preprint now that's coming out, and it's in Press and GigaScience if you want to have a look at it, that talks about how you organize your metadata, the kind of information that people collect. And it has like tables and guidance on the different fields, what information you can collect and what information is required for public submission to the different repositories out there. So that's a preprint there you can have a look at. And it goes into a lot of detail and gives you a lot of guidance in that space. One thing is to always implement a system that will try to help you ensure constant metadata. Try to move away from just sending emails around with different spreadsheets and try to have a more complex system. It can be, depending on how much infrastructure and how much effort you want to put into it, it can be something like spreadsheets on a shared drive in the cloud to a huge website or API application. And if you're interested in an API application, you can have a look at the Majora thing that was developed through COG, and that was what we've been using for what that might possibly look like. But if you're confident with Python, these are a bunch of Python libraries I've used. So Marshmallow, PyExcel for reading Excel files, or Pandas as well can do it, and GSpread for using Google Sheets. These are things that are very useful that I use every day that helps me sort of corral metadata and produce reports and so on. Using a good sample identifier is a great start. You do have to keep in mind that your samples are going to be out there, and hopefully you're going to put them out quickly into the public space so other people can use them. And you do need to think carefully about the sample identifier. The critical thing is it shouldn't be patient identifiable, and that might mean that you just, as a matter of habit, you take whatever identifier that you've been sent and just give it your own one, just to make sure that it is truly anonymous. But other than that, you want to have an identifier that's simple, ideally alphanumeric, so it's easily readable by a computer. You don't want strange characters in it, and you want something that's easy to describe, something you can talk to another human being about without causing confusion. And again, there's, I think in the Majora preprint, we go into what COG was using for designing their barcode, and that's one place, another resource you can look to for guidance in that regard. So this is the last snippet from me. Data submission is really, really important. By sequencing SARS-CoV-2, you're contributing to a global effort. Your data can be used by others to hopefully, get us out of this. I mean, you should deposit the raw data and genome sequences in public archives. The main ones are GISAID, which takes the consensus sequence only. It's much easier to use. And I'll give a little secret. If you do want to get in touch with the curators, like you want to update samples you've already submission or you have any queries, here's the email. It's not readily available on the website, so this is a little secret to get in touch with them directly, and usually they get back to you in a couple of days if you contact them through that email address. For the INSDC, so your EBI and CBI and DDDBJ, that takes reads and consensus sequence. It's more complicated because it's structured hierarchically. So biosample has many reads attached to it, which has many assemblies attached to it. It's a lot more confusing to get used to. But in both cases, it's really important to explore how you submit and get registered on all of these repositories before you're at crunch time and you need to submit your data for publication, and it's due yesterday. That's not the time to start. You should try and register and try these submission systems out beforehand, because they can be a little bit complicated. And again, if you look at the preprint I gave you for guidance of what fields are required for the different repositories, that'll also help you a lot with understanding what you need to provide. So here's all the readings I mentioned throughout the talk. You can go back to them later, and I'll sort of post these slides somewhere so you can also have a look. But yeah, that's it from me. And I'll take questions, I guess, at the end in the discussion panel.
Thanks, Nabil. That was great, short and sweet. So I'm going to give people a chance to answer questions now because we are kind of ahead of time in the chat. But while people are thinking of some questions, I would. So there was a previous question actually in the first session, which was really about saying, well, what is the kind of extent of thank you, Andrew. What is the kind of extent of problems in GISAID, particularly relating to kind of contaminated sequence? And we kind of decided that we couldn't really estimate that very easily. What do you think the future looks like in terms of databasing? You know, this kind of mixed economy where we have GISAID for consensus and ENA for reads. Is that the kind of stable model? Can you see a future model? And how can we kind of either prospectively or retrospectively deal with some of the quality issues in the global databases? Nabil, I've lost you somehow. Oh no, you're there. Good. But you're muted, I think.
You're mute.
Hello.
Hello. Yeah.
That's better. Something went wrong. Okay.
Where are we going with the repositories? I think I've definitely seen a lot of improvements in the services ENA and CBI have provided, and they are slowly catching up. And I think at some point they will probably be the primary archive for data, as they are for many other organisms. I haven't seen anyone come up with anything outside of GISAID or the INSDC. And but GISAID, I think, is still going to have, GISAID has this advantage of being this ease of use. It is really easy to jump on and start submitting your data, and it is the primary resource, but things like NextTrain and so on are pulling from. So it's going to be business as usual, I think, Nick. Maybe hopefully INSDC can make an offering that we can switch to. In terms of data, we've run into, I think, you know this as well, like we've run into so many issues where having just the consensus sequence is not enough, and having the raw reads to go back to for issues around quality control or going back and doing more advanced analysis is really, really important. And I think that that's going to become more of a requirement, because now people are going to start doing more research-based questions. A lot of people are going to start trying to take COVID genomics and human genomics and start putting that together, and having the raw reads is really, I think, critical for that kind of research question. So yeah, I think we're going to go back to using GenBank for all of our stuff, hopefully with these submission pathways, but yeah.
Okay. Oh, that's an interesting perspective. Yeah, I mean, I guess that would be nice in certain ways. I mean, I don't know. It'd be interesting to hear from people in the audience what their experiences with the data deposition. If anyone wants to contribute a comment or a question, But I guess my observation has been that GISAID has probably become popular just by virtue of how easy it is to submit data, perhaps relative to getting into the INSDC database, and also how quickly data is published online, within a few hours often of being uploaded, and it's difficult to do that in GenBank at the moment. And maybe that's the reason, and I think this kind of follows on nicely with the question that Andrew has pasted on the chat, which is, what do you think about Deltacron? I mean, if that was for those not following Deltacron, that was a case where some sequences were posted on GISAID indicating a hybrid variant with mostly, I think, I can't remember which way around, mostly Delta sequences, a bit of Omicron, or no, mostly Omicron sequences, a bit of Delta in, I think. And of course, it goes from GISAID to Twitter to the front pages of newspapers within about 2 days, without really any kind of ability to step in and say, well, hang on, is this real? Kind of the sort of peer review idea, either just informal peer review on social media or formal peer review through the kind of journal system. And suddenly, you know, everyone's kind of scrabbling to prepare reports about what this might mean, what it might not mean. And actually, you know, that's a good example of something that really comes down to probably data quality issues. This is also setting up very nicely for the next talk, I think. But I mean, what do you think about that, Nabil, in terms of what do you, when you see things like Delta Cron, what do you think? And also, how can we kind of mitigate?
Yeah, I mean, because it's only the consensus sequence, it's difficult to unpack exactly what went wrong there. But it is, I think the consensus is that it's actually just kind of one of the problems I was talking about, cross-contamination. There's a delta on one plate and Omicron and just made a mistake somewhere. And that's what creates it. And that's how bad it can get. I think what's going to happen is hopefully we're moving away from where we started with this, which was panic, right? We started this pandemic, we're like, we need to start sequencing, we need to start getting a handle on what's going on. And now we're going back and asking these quality control questions. And I think people are going to look at these examples and start saying, well, this is down to the fact that there's not enough due diligence or there's not enough transparency with the data provided in the way it's provided. So we need to do a better job. And I think, yeah, we're going to change our MO to allow for that a bit better.
Yeah, I think you're right. A lot of this is down to just moving so quickly, the general level of interest, you know, the need to react. But as you say, we're kind of probably moving into more of a business as usual phase, more generic, more general surveillance approach. And it would
be living with COVID.
Living with COVID, yeah. And I really think for me, I think you alluded to this really, is the ability to properly link consensus with the underlying raw data would be a huge advantage. It's something we can do in the UK, as you say, because on Climb, we were able to store those data sets close to each other with an analysis platform, which is very helpful. But internationally, it's much more patchy, the sharing of the underlying data and the linkage between the consensus in GISAID and the reads over in INSDC. So that feels like an obvious thing to improve. Okay, so there's a question in the chat from Amir. Thank you for this excellent presentation. So with the high error rate of Nanopore compared to Illumina, how can we be sure that mutations detected by Nanopore, which is critical in designation of new variants?
I'm sure Josh covered this, right? Why this doesn't matter?
We didn't really cover that specific question, although I think what Josh did present very effectively was that actually a lot of the variability in results can be generated by the choice of targeting, you know, amplification method and sample and library preparation methods are often sources of variability in the data, kind of outwith the actual choice of the sequencing platform. But I guess this question is particularly about, you know, Nanopore error rate, which obviously as a single molecule technique is higher than Illumina, although I think most people would agree that the error rates come down significantly over time. But and it also speaks to the discussion around how consensus is built. You don't build a consensus from a single read, you build from an ensemble of reads, you know, typically 20-fold at least, but usually hundreds of fold coverage. And I guess in our experience, mostly that resolves issues around sequencing quality, except for very occasional problems in very difficult to cool parts of the genome, such as long homopolymers have always been a bit of an Achilles heel of the Oxford Nanopore platform, although thankfully in SARS-CoV-2 there's very little in the way of long homopolymeric tracks. But would you agree with that? Was there anything you'd say about that? You did a study on this, didn't you?
I think I had the same question when we started, and now I convinced myself that it's fine, and I've forgotten. I sort of just live with it now. We've been doing it so much; it's never really become a problem. And the reason is that the error rate in a nanopore is stochastic. That's it. We remove the error because we sequence so deeply. So it all filters out. It all works out in the end, basically. And it hasn't been a concern, despite having done whatever million of sequences we've done.
Yeah. I mean, it doesn't seem like that's probably the a major source of error, really. The major source of error that and this is, segue me excellent, very well into the next talk. It has been this issue around laboratory handling samples, particularly in combination with what Josh talked about, which is these dropouts that occur in amplicon sequencing, where particular regions don't sequence at all. Then any kind of basal level of contamination, index hopping, problems like that tend to become apparent because they fill in those gaps where there should be no sequence. There's a little bit of sequence, and that seems to be, that generates your problem. That's probably the story of Deltacron, and that's probably the major source of error, other than the other source of error that Sam Wilkinson talked about, which is about improper bioinformatics handling. And you also mentioned, Nabil, using the wrong technique or the wrongly configured bioinformatics technique for this particular type of data. That's kind of why we're laboring the point, because this is a particular type of data that's distinct from whole genome sequencing, random genome sequencing.
So basically, it's like, this is the least of your worries. The error rate of the sequencing platform itself. You've got bigger things to worry about. I think something that's more of a headache is when people put the wrong collection date more than anything else. And then you start saying Omicron appeared in July last year, and you're like, that doesn't make any sense.
Yeah, these metadata issues are a whole other, and you also mentioned those in your talk.
Yeah.
Okay, well, thank you very much.